you so much for the kind introduction, and it's great to be here in Cairo. It's like Dr. Amir said, it feels like home. So let me. So the topic of my talk is how to come up and improve Treg cell therapies for transplantation and autoimmune diseases. And I will present to you some background and some of the work that we have done. So before I start with Treg cell therapy, first I should say what is a Treg. And essentially, if you think about the Treg, it's like the police of the immune system. So every time, for example, we get a pneumonia, our immune system has to fight the bacteria or the virus, whatever we're infected with. But this response has to be contained. Once the pathogenic virus or bacteria is actually killed, our immune response has to stop. Because if it doesn't stop, this is gonna destroy us. So the immune system has to stop. And one of the ways, probably the most potent cell that is responsible for stopping this immune response is the regulatory T cell. So it's a subset of CD4 cells that actually is tasked with tempering immune responses. And it can do this by two ways. One is the T cells can engage other cells and stop them, or they can secrete certain molecules to actually stop the immune response this way. So either contact-dependent immunosuppression or contact-independent through soluble factors. And you can see some of them here. Now, the question then becomes, how important are Tregs for our life, our existence? And what I'm going to say is that Tregs are actually indispensable for life. If we do not have this cellular population, then we will die. And I will start first with the example of mice. So FOXP3 protein is actually a protein that is actually highly expressed in regulatory T cells. And this protein defines regulatory T cells. So when mice are actually born without FOXP3, which means that they do not have any regulatory T cells, as you see, they die within 3 weeks of age. And the reason why they die is because their immune system essentially infiltrates all of their organs. So as you can see, the size of the mouse is much smaller because these mice die from really bad enteropathy. So they have a cholera type of syndrome. Their skin is actually infiltrated by all these immune cells. So it's a type of an aggressive lymphoma that actually pretty much kills the mice. And that's actually true not only for mice, but also for humans. And because the FOXP3 gene is actually located at the X chromosome, men are way more prone to develop this disease that it's called IPEX than women. And what is this IPEX syndrome? Essentially, IPEX syndrome is the similar syndrome to what is happening in mice. It happens in men, people. And essentially, you have this uncontrolled T cell activation, and these T cells go and attack the intestines, so you have a perfuse diarrhea, and they attack almost all of our endocrine tissues, the stomach, the kidneys, of course, and then essentially it's a fatal disease. Unless you do a bone marrow transplant and you reconstitute the T cell compartment with Tregs, these boys will universally die. Even immunosuppression will not do much. As you will see here, all of these boys will be dead by 25 years of age, even with immunosuppression. So the only hope is actually bone marrow transplant. So Tregs are important for life, but then how important are they in very common diseases that humans suffer from, such as cancers, infections, and autoimmune diseases? Well, one thing I can tell you for sure is that they are actually avidly present in all of these diseases. As you can see, this breast cancer tissue here, it's actually full of Tregs. The green color is the Tregs. As you see in the middle, in the case of leishmaniasis, there is a lot of Tregs that are actually colored brown. And of course, in autoimmune diseases, you can see the Tregs lighting up as red, and they are surrounding an islet, an insulin islet, and trying to prevent the attack by the blue CD4 cells. So Tregs are important for life, and they are involved in many common human diseases. So then, essentially, the FOXP3 protein was discovered about 20 years ago. It's a fairly recent discovery. And since that time, researchers started thinking about this possibility that we can maybe use these regulatory T cells to treat human diseases, such as transplant rejection and autoimmune diseases. So in this type of concept, what you do is you take a blood unit, you isolate Tregs from there, you grow them in the laboratory. This is actually a picture of Tregs growing in the laboratory, and then you give them back to the patient. And with the advent of gene editing techniques, now not only you can give Tregs, just polyclonal Tregs, but you can actually engineer them. You can engineer their specificity, you can make them CAR, you can make them TCR specific, you can give them genes that you want, you can take out genes that you don't want, and then give them back. And I think that's actually something that holds a promise for the future. So this idea of Tregs giving to patients has actually been trialed, and probably the biggest trial today is the one study, which essentially it's 7 studies altogether, and it was a multi-nation study, very many different centers from around the world, and essentially what they did here is they took unsensitized kidney transplant patients, and instead of giving basiliximab induction, they gave, in most of the cases, regulatory T cells. So before the transplant, it was living donor transplant, of course. So they actually expanded the Tregs, they gave the kidney, they gave the Tregs, and they actually did not give any other induction, and they kept going with standard immunosuppression, although the patients that received the cell therapy induction, as you can see there, 60 weeks later, they were only maintained on one medication, tacrolimus. So the big question here was, is this cell therapy induction going to be able to prevent rejection the same way basiliximab does? And the answer is yes. So cell therapy, polyclonal Tregs were able to prevent rejection at the same rate. And the huge benefit here, as you can see, is not only they prevented rejection, but we had way less infections. So where do we go from here? Number one, well, Tregs are safe. So there have been many trials. We have never seen any adverse effect with Tregs. So number one, they're safe. Number two is, well, we started by giving them to unsensitized patients, which is actually an easy task, right? Well, I shouldn't say easy, but it's where we start. Now we probably have to think about how are we going to give these cells to rejection? How are we going to give these cells to sensitized patients? So now we have to fortify them away. So probably polyclonal Tregs will not be enough to hold a more active immune system. They have to be fortified to be able to fight. So that's the one thing. And the second thing is, we saw from the first trials that we were not able to grow these Tregs too much. And we only have 5 liters of blood. And obviously we cannot remove the whole blood because the patient would be dead. So we can only remove a certain number of Tregs to start. So what we need to improve is the expansion rate in the ex vivo culture. So if, let's say, we start with 5 million cells, we have to be able to grow them so that this therapy will be feasible. Because if they don't grow very well, that's not going to be feasible. And that was actually one of the problems that the first trials encountered. So this is now our contribution to the field. And essentially, the same time that the clinical trials, these first clinical trials were going on, we were in the lab and we were trying to see how we're going to grow the cells. And just to give you an idea of what was going on here, on the panel A and C, you just see Tregs growing. And this is a picture of day 8. So you take these cells in culture, you grow them, and then by day 8, you've gone through a full cycle, and these cells should be essentially kind of like what you see here. They should relax. So they go, it's kind of like a heart cycle. Like it goes through systole and diastole. So by day 8, they relax, they're ready to go. As you can see, when you start giving these cells inflammatory cytokines, you see these black dots. These black dots are actually activated segregates, clusters of Tregs. So that actually gives you a pretty good idea that these cells now are getting more activated. So the and it kind of makes sense, right? Because when you have a regulatory cell, you want the cell to be able to scale to inflammation, to sense inflammation and say, hey, there is inflammation here. So maybe I should grow more. I should work better to actually be able to fight it. So then we tried this different activation methods and different cytokines. And as you can see, just one slide that you can see in the one condition that you can see here, we were able to take the cells from the standard way of growing around 20 to 30 times with one cycle of stimulation. We were able to get this number to go up to 1,000 times. And this was after we changed the mode of stimulation and we gave the Tregs IL-6 and TNF-alpha. So we got the cells to grow a lot. But were the cells that we grew a lot good cells? And since we were pushing these cells with inflammation, we wanted to see what type of receptors these cells have to essentially sense this inflammation. And through a CRISPR experiment here, we identified that TNFR2 is actually the receptor that helps the Treg sense how much inflammation is around so that it can actually grow more. When we deleted this gene with CRISPR technology, we actually lost all this proliferative effect. And of course, like I said, we needed to make sure that when we pushed these Tregs to grow more, that they were still Tregs. So we checked their FOXP3 expression, Helios expression, and demethylation status. And as you can see, they maintained a very good Treg status even after all this expansion and exposure to cytokines. Now, after, obviously, one of our first goals was to be able to improve the ex vivo expansion technique. So in order for this therapy to be widely applicable and to be able to treat patients, you have to make sure that when you start with 5 million or 10 million Tregs, you have to be able to get to the billion type of number so that this is an enough dose for a human being to be treated. Because if you treat any human with 1 million cells, I mean, that's probably not going to do anything, even if these cells were so potent. Just to give you an example, when we actually treated diabetes in mice with these cells, we actually still needed about 1 million cells to get delay of diabetes. So if you calculate per weight or per CD4 count, you have now to go up to the 3 billion range so that a human will be appropriately at least dosed. If we actually kind of think that there is a linear scale between a mouse of 25 grams to a human of 75 kilograms. So with this, we filed this patent application to essentially say that we identified a new method to grow these cells. And what did we do with this? As you can see, the blue charts are the standard clinical trial method. So what happens is you give the Treg a bead that has CD3 and CD28, which essentially it's the common way of stimulation of any cell. So you tell the cell, here is an antigen, here is co-stimulation, just go and grow. And you provide it with IL-2, because T cells need IL-2. And that's how we grow cells, actually, T cells in the clinical trials. And as you can see, you can get a wide range of results. Some people's T cells grow 50 times, and some other people T cells go and grow 1,000 times. And it's all over the place. So you can't really run a clinical trial reliably, waiting for 25% of the patients to grow 20 times and the other ones to grow 1,000 times. So with our method, which is essentially the final method, is the one that is all the way to the right, as you can see, we were able, by switching the mode of stimulation and providing these cytokines, to get almost consistently above 1,000-fold of expansion. So this essentially was our contribution, first contribution to this field. We filed this in 2020, where we go from here is actually we want to identify how these Tregs essentially work and what type of genetic modifications we need to introduce so that these cells can track to the sites of inflammation. So we can give them through the blood, and they can go to the kidney, they can go to the lymph nodes, they can go to the heart, they can go wherever we want. So one thing is how we track these cells. The second is how we actually make these cells work better so that they don't really rely on molecules that are actually produced in the human body. So they can actually, by themselves, kind of feel that there is a lot of inflammation. So this way they can actually work more. And then obviously we have to introduce a switch there so that we can actually turn them off once the treatment is over. So essentially we're working now on genetic modifications. And yesterday you were touching a lot on epigenetics, and epigenetics is actually something that we're really investigating. Obviously with epigenetics, you can actually hit a transcription factor, but then you would have a wide range of impact. So it makes it easier from the perspective that usually these drugs are cheaper, but it is harder from the perspective that you have a pretty big impact on the cell. So if you want to be more targeted, then what we do is now we actually do epigenetic screens, and we try to figure out what particular proteins are affected by the transcription factors that we are actually seeing different in these cells. So this is work that is currently ongoing, and I hope that during one of the next years, we can actually discuss more about the epigenetic modifications that we're actually doing. So I would like to thank you so much.